Hi, I'm Dongwen Lyu. I'm an assistant professor at the Georgia Cancer Center. I'm a molecular and chemical biologist, and my lab focused on developing small molecule degraders and modulators to treat cancer and different diseases. Because in US or even in the whole world, a lot of people still suffer from different kinds of cancer. Proteolysis targeting chimera or PROTACs. It's a new modality can induce protein degradation through recruiting the E3 ligands such as CRB or VHL. PROTACs have emerged as a new technology can degrade the target disease-related target protein, especially previous considered undruggable proteins or difficult drug protein by the conventional small molecule inhibitors. The small molecule binder is it's just like a chemical molecule. It can bind to the protein, not exactly inhibit the protein. Sometimes it just binds there. And when we generate PROTAC, pure PROTAC is a molecule. It can recruit E3 ligands to induce protein degradation. Currently, more than 40 PROTACs are in clinical development. However, a persistent bottleneck is that many disease-related proteins lack trackable binder, to generate final PROTAC molecule. Discovering such binder is super difficult. It's like a time and resource intensive. So especially for the targets without obvious ligandable pockets. In my lab currently, I'm working on the leukemia, breast cancer, and prostate cancer. But as you know, this kind of protein degradation technology, it can be used in any kind of disease because our target they are proteins, right? If the protein play essential function in that disease, we can use our degradation system to degrade that protein, to achieve maybe potential therapeutics. In our recent published work, we report a new platform named polyhistag that can selectively degrade polyhistidine-tagged proteins by recruiting either the CRB or VHL E3 ligands. It provides a convenient tool for degradability assessment. The polyhistag, such as 6-histag, is among the smallest and easiest epitopes that can be seamlessly inserted at the gene terminus by CRISPR-mediated gene editing. In addition, the system is also valuable in basic research because polyhistag is minimal and it reduces the risk for perturbing native protein function. In the same time, it can enable controlled protein degradation, which is particularly useful for studying protein function. So we want to use small molecule, to treat different disease-related proteins to find a way, to maybe cure the patient or release, the pain or the difficult time for the patient.